Hello friends, in this video we are going to discuss about eukaryotic DNA replication in brief, or we will see the overview of eukaryotic DNA replication. It is DNA duplication process so that exact copy of DNA is received by daughter cell. And the site of DNA replication is nucleus. Features of DNA replication, it is semi-conservative. One is the parental strand or the template strand and the other is the newly synthesized strand, bidirectional. This also we have discussed earlier. You can refer my video, the link is given in the description box. Here from each origin, two replication fork proceeds away from origin in opposite direction, hence it is bidirectional. It is semi-discontinuous because one strand is synthesized continuously, known as the leading strand, and the other strand is synthesized discontinuously in the form of small fragments known as Okazaki fragments, and this strand is known as the lagging strand. Origin of replication, it has multiple origin of replication, hence it is multi-repliconic, as each of the origin defines a replicon. What is replicon? It is the stretch of the DNA that is replicated from a particular origin. This also we have already discussed, and why it has multiple origin of replication: in order to finish replication of larger size of eukaryotic chromosome on time. So eukaryotic chromosome has larger size compared to the prokaryotic DNA, and replication should be completed on time before the cell divides or before the cell division. Hence, it has multiple origin of replication. Now, let us see events that is related to replication during cell cycle. So, in cell cycle, we have discussed that there are four phases: G1 phase, S phase, G2 phase and M phase. In the G1 phase, DNA replication regulatory process are initiated. In S phase, duplication of the genome takes place. Vast majority of DNA synthesis occur. So it is known as the synthetic phase because mainly the synthesis of the DNA takes place in this phase. G2 phase, damage or replication error is corrected. So during the DNA replication, if any error occurs, it is corrected during the G2 phase, and also any damage that occurs in the DNA due to mutation or ultraviolet radiation, it is also corrected during the G2 phase. In M phase, one copy of genome is segregated to each daughter cell after cell division. So eukaryotic genomes are larger and more complex than prokaryotes. Eukaryotic chromosome is linear, while in prokaryotes, in E. coli, we have seen that it has circular DNA. And in eukaryotes, DNA is bound to histone proteins. Hence, DNA replication is also very complicated in eukaryotes, and it involves several enzymes and other proteins. There are three stages or steps of DNA replication: initiation, elongation, termination. We will discuss these three steps separately in detail in series of videos. Here we will see the overview or summary of these three steps. Replication begins at origin of replication. In eukaryotes, there are multiple origin of replication. These origins are recognized and bound by certain proteins known as origin recognition complex. This ORC or origin recognition complex then recruits other proteins, including two copies of helicase enzyme. These helicase enzyme are present in the form of hexamer MCM2 to MCM7. Helicase enzyme then unwind double-stranded DNA using energy in the form of ATP, as a result of which two replication forks are formed at each origin, and these replication fork then extends in opposite direction. Hence, two replication fork moving in opposite direction creates replication bubble. Because there are multiple origin, hence multiple replication bubbles are created. As replication fork proceeds, supercoiling occurs. This we have already discussed in detail in separate video. You can refer my video, the link is given in the description box. This supercoiling is then relieved by topoisomerase enzyme. We have already discussed this topoisomerase enzyme, you can refer it, as a result of which single strands of the DNA are produced, and These single strands of the DNA is stabilized by RPA, which is protein, and it is similar to single strand binding protein in prokaryotes. And binding of RPA then prevents re-annealing of two strands of the DNA. Then the priming occurs, or the synthesis of the primer. Primer is synthesized by DNA polymerase alpha. It synthesizes RNA primer, which is 10 nucleotide long, which is followed by the synthesis of short stretch of DNA, which is less than 15 nucleotide long. But because of its low processivity, DNA polymerase alpha cannot synthesize or extend the new strand of DNA. Hence, this DNA polymerase alpha is replaced by other DNA polymerase. In this case, DNA polymerase delta or DNA polymerase epsilon, which are the main replicating enzyme for the synthesis of complementary strands. And these synthesize the new strands in the direction of 5' to 3'. We have already discussed in eukaryotic DNA polymerase that if one strand is synthesized by DNA polymerase delta, then the other strand is synthesized by DNA polymerase epsilon. Mostly, DNA polymerase epsilon synthesizes the leading strand and DNA polymerase delta synthesizes the lagging strand. But it is still not very clear and research is still going on. So after the strand synthesis, primer is removed by RNase H and FEN1 and replaced with new DNA nucleotides. But nick is present between the DNA chains. This is sealed by DNA ligase enzyme. DNA ligase enzyme we have discussed in detail in separate video, you can refer it. After the completion of this DNA replication process, two sister chromatids are obtained, which are then transferred to each daughter cell. Since eukaryotes have linear chromosome, so there is problem in the replication of ends of the chromosome known as telomere. This is replicated by telomerase enzyme. This we will discuss in detail in separate video on termination. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.